Okay, we are back to the MN blood group. So again, codominant. Both of them are going to express, and that gives you this middle M and N together. Incomplete dominance, intermediate blended phenotype, right? Which is the very definition of codominant. Both of them are dominant. Okay. Multiple alleles. Sometimes 3 or more alleles of the same gene can result in a unique pattern of inheritance. This is our chinchillas. This is our ABO blood group because there's 3 genes. There's A, there's B, there's O. There's 3 things that can mingle to give you a blood type. A and B antigens are present on the surface of red blood cells, but there are 3 alleles, A, B, and O. And we talked about this. This is just a review. Some people do them with I's. I just do them with O's. It doesn't, you can do it either way as long as you keep it straight. And this is again the antigen, the phenotypes, the 3 alleles, the A, the E, the O. A and B allele produce respective antigens. The I or the O allele does not produce an antigen. A and B are codominant. A and B are dominant to I or O, and A and B are codominant to each other. Does that make sense? Take a second. Pause the video. Does this make sense? A and B are both dominant to O or I, whatever you want to use. If it's AO, you have blood type A. But if it's AB, those are codominant to each other. So take a second, make sure that makes sense. Okay, Bombay blood type. Here we go. The Bombay phenotype. So this is kind of like being an albino where it doesn't matter what the gene says because you don't have any pigmentation. Both A and B antigens are derived from a precursor molecule, H substance. The Bombay phenotype has an incomplete form of that substance. So it's a rare recessive mutation. So results in an inadequate substance for an enzyme due to a rare mutation. What does this cause? It causes type O, you to have type O blood, regardless of what your parent's genotype is. So somebody has a type O, one parent has a B, and the female is B, we don't know, allele to 2 children. The female homozygous for the FUTO allele prevents her from producing that H substance. So no substrate to make an A or B antigen. So her ABO blood behaves as a type O, yet genetically she's a type B. Wrap your mind around that for a second. So the Bombay blood type trumps, doesn't matter what your real genetic blood type is, because you're not making an antigen anyway. Because the FUT1 gene doesn't allow your body to make an antigen. So it sort of masks the AB blood type. So you can be a type A, a type B, but you're not going to behave that way. You're going to be phenotypically an O. This really helps to explain when parents are not, you have questions. You can say, oh, it could be the Bombay blood type. A partial pedigree of a woman with a Bombay phenotype. Functionally, her ABO group behaves as type O. Genetically, she is type B. So we've got an A and a B as the parents. You shouldn't have a type O here, but you do because she has the Bombay blood type. And then when you mate those two, you get an actual phenotype here. She didn't pass on the Bombay blood type. She passed on her actual blood type as being a B. So that's just something to think about. There is a recessive mutation that causes white eyes in Drosophila, and it was discovered in 1912, and this means that eye color ranges and an absence of pigmentation. And we will probably maybe do a problem in which there is a white eye problem. Let's talk about horses. I feel like we did this, Cremello and Palomino. Here's the different kinds of horses. Chestnut is brown. Palomino is that real light brown color, and this is the same thing. This horse actually has some of both of these hairs. It's producing both. However, Chestnut is dominant to Palomino. This is just both of them together. So here's just, if you went ahead and did the crosses there, that's what you would get. From the results determine the mode of inheritance. So this is essentially a co-dominant situation. Here is what I was talking about with the pedigree for hemophilia. So here you have Albert and Victoria in the late to early 19th century. They had another Victoria, Edward, Alice, a no-name child, Helen, Leopold, and Beatrice. Okay, oh no, that's good. We'll stay over here. Okay, now all of a sudden, Leopold had hemophilia. Well, that's not good. And then it gets even stranger, in which Frederick all of a sudden has hemophilia. And all the way over here, Maurice has hemophilia. So you can tell what's happening, and the culprit was Queen Victoria, in fact, had hemophilia and gave that to some of her children. Who is believed the original character? Analyze the pedigree and indicate which females are also certain to be carriers. What is the probability that Princess Irene is a carrier? So Victoria is a carrier. She doesn't have the disease. If she had it, then all of her male children would have it, right? She doesn't have it, but she is a carrier, and that's where you get her passing it on to Leopold, who probably has a wife somewhere who's also a carrier, probably both. A little something about lethal alleles. Lethal alleles are essential genes. So this is like the Bombay blood type. That was a lethal allele. You don't have any blood type if you have not producing antigens. So these are required, but lethal alleles go a step further, and they are required for the organism's survival. Mutations resulting in non-functional gene products can be tolerated if heterozygous. So one wild type allele is sufficient for survival, but the homozygous recessive will not survive. This is sometimes what you'll see in pregnancies or miscarriages, or just a person struggling to carry a child. Or in fact, it's not a viable pregnancy. It's not even a viable, once the embryo starts to grow, it's a lethal allele. Recessive lethal alleles result in homozygous recessive individuals and do not survive. Homozygous lethal alleles may result in a distinctive mutant phenotype in which there are no homozygous recessives. Like, they don't exist. You're like, what is happening? What's happening is it's a lethal allele. So if you have like little a, you don't survive. So you don't actually, the embryo can't grow. So you will see this just again when you're looking at pedigree. So be like, what is going on? That's what's going on. Mutation in mice causes yellow coat color, varies from normal agouti coat phenotype. Crosses between two strains lead to several phenotypes. Homozygous yellow coats are lethal in mice. Mice die before birth. There we go. So you have the agouti mice color, right? Yellow allele, YY. Mutant allele, Y. Behaves dominantly to normal allele coat color. Behaves as homozygous recessive, and it's lethal. The genotype AY, the agouti, the yellow, two yellows, and you do not survive. So this very often happens when you have two agoutis that make little nice babies. A dominant lethal allele is Huntington disease. We talked about that a little bit in class. It doesn't really count as a lethal allele all the way because you are born and you do have the disease. It just kills you around the age of 40, and it is dominant. So that is what's very strange about it. It's dominant, but you've already had kids by the time you survive into adulthood until 40, and then you find out you have Huntington's disease. Some more modifications. We'll do that in the next video. We will talk about different modes of inheritance. So we already talked about blood type. We talked about albinoism, or we will talk about albinoism, and we'll talk about anything that does not a 9 to 3 or 3 to 1.